Today, we will go over how to thaw human dermal fibroblasts. You will need cell media. Our cell media includes 10% fetal bovine serum and 1% penicillin streptomycin. This is cell dependent. A 50 ml centrifuge tube and a 15 ml centrifuge tube are also needed. We will use a T75 flask to grow the cells. The T stands for tissue culture treated polystyrene, and the 75 stands for 75 centimeters squared. Both a autopipetter and a micropipetter are required for this task. First, transfer 50 milliliters of HDF cell media into a 50 milliliter centrifuge tube. This makes it much faster to heat up the media and have it exposed to less air. Label the tube with HDF cell media, the date, your lab, and your initials. Place the cell media into the 37 degrees Celsius water bath. The cells that we will be using are located in the negative 80 degree freezer. They are on the top shelf in a box. The cells are inside cryotubes. This preserves them during the freezing process. The cells are labeled with their cell line, the passage, the amount of cells, and the date. Slowly warm up the cells by twisting them in between your fingers. Add 3 milliliters of HDF media into the 15 milliliter centrifuge tube. Add all of the cells into the 15 milliliter centrifuge tube. Once the cells are added, we will need to centrifuge the tube. To centrifuge the tube, turn the centrifuge on by the switch in the back. To use the centrifuge, make sure you have 2 tubes with the same amount of liquid in it, and add them on either ends of the centrifuge. This balances the centrifuge. For this process, we will set the centrifuge tube to 180 Gs and to 7 minutes. Press the play button to begin centrifuging. To open the T75 cell flask, open the corner and then spread over the top. Today we will be using 2 cell flasks. Close the bag by pinching the 2 ends together into the middle and then rolling together. Make sure to tape the bag shut and label with the date. Label the flask with your cell line, the passage number, your PI's name, your initials, and the date. Once done with centrifuging, there should be a small white pellet at the bottom of the centrifuge tube. Carefully aspirate out all the liquid inside the tube. Be very careful to avoid the cell pellet at the bottom. Next, add 2 milliliters of cell media into the 15 milliliter centrifuge tube. Now mix the cell pellet into the media. Do this by slowly sucking up the cell pellet at the bottom and then injecting it back in with the micropipetter. Mix thoroughly so that there are no visible cells left. We will now add the cells into the T75 cell flask. To open up the flask on the left first, and then the one on the right. Do this to avoid going over any of the lids or the bottles. Add 1 ml of cell media mix into the cell flask. Mix the media a couple times in the 15 milliliter centrifuge tube before adding to the flask to get an even number of cells into each. Add 12 milliliters of media into each flask. This will provide nutrients to the cells. The amount of media used is dependent on the size of the cell flask. Be careful not to let the tip of the pipette touch anything on the flask. Just let it hover inside the opening and then pour in the media. Close the flasks in the reverse order of opening them. Be very careful not to move over the open flask. Slowly tip the flask forward and oscillate from side to side to get the media to cover the entire bottom. Place the cells into the incubator. The incubator needs to be set at 37 degrees Celsius and 5% CO2. This helps the cells grow.